Welcome to class. Today we will be learning about carnivorous plants. Before we get started, obey your curiosity. Like, subscribe, and click the class bell to be alerted every time a new lecture is posted. Today's lecture is separated into 6 major sections: the overview of the subject, followed by defining characteristics, wherein the trap types are elucidated. Then the manner of digestion is discussed. Following, we will walk through geological time while learning about the evolution of these amazing plants. Nearing the end of the lecture, we learn about carnivory and angiosperms generally. Lastly, our lecture ends with a call to action about conservation threats and the conclusion of the lesson. Let us begin with appetite, the kind that both fills the belly and that which feeds understanding. Carnivorous plants are the scholars of deprivation, creatures of chlorophyll that chose hunger as their tutor. They grow where life often does not, in waterlogged light, in soils too thin, too poor in nitrogen for ordinary ambition. And so they learned to hunt. From moss to marsh, and even the desert, they stretch their green intelligence toward motion. They lure, they trap, they consume. Not out of cruelty, but adaptation, an elegant calculus of necessity. Charles Darwin, ever the student of strange hungers, published Insectivorous Plants in 1875, giving these quiet predators their due. He recognized that intellect does not always wear a nervous system. Sometimes it simply grows teeth where others grow leaves. True carnivory has evolved at least 12 separate times across 5 orders of flowering plants. An evolutionary refrain: when the world withholds, creation learns to take. Today, we know of more than 800 species that live by this lesson: Venus flytraps, pitcher plants, bladderworts, each a variation on the same refrain: attraction, capture, digestion, assimilation. Even now, more than 300 proto-carnivorous species hover at the edge of hunger, half committed to the art of consumption, half still pretending to be vegetarian. And, as with all intellect, scarcity and extinction shadow their brilliance. As of 2020, 1/4 of known species are threatened, another demonstration that curiosity, too, must fight to survive. So remember this: evolution rewards not the gentle, but the curious, the adaptable. To think is to trap. To learn is to digest. And so, let us consider what makes a plant a carnivore. Plants are considered carnivorous if they display one or more of these 5 traits. 1. They capture prey with specialized morphological structures. 2. They subdue or kill the captured prey. 3. They digest the captured organism via enzymatic digestion or bacterial action. They assimilate the resulting nutrients, and then utilize those nutrients for growth, repair, or reproduction. Notably, Other traits may include the attraction and retention of prey. There are 5 fundamental mechanisms by which plants transgress their verdant passivity and cross into predation. First, the pitfall trap, the picture of doom. A leaf folded upon itself becomes a chamber. Nectar lines the rim, drawing the unsuspecting to the edge of annihilation. They fall, and the enzymes, or sometimes entire ecosystems of bacteria, complete what the leaf began. Second, the flypaper trap, adhesive appetite. Here, the leaf does not close, it holds. Mucilage, clear and glistening, betrays its gentleness. The prey struggles, and in so doing quickens its own dissolution. Third, the snap trap, jaws of the plant kingdom. Speed is this evolutionary boast. A Venus flytrap senses the trembling of its prey and folds upon it in a gesture almost animal. Fourth, the bladder trap, vacuum in action. Invisible to the naked eye, yet fierce, this is the domain of Utricularia. A vacuum forms within the bladder. The door is baited, sensitive. A single brush and the prey is drawn inward by suction, as if the plant inhales thought itself. And fifth, the lobster pot trap, a patient spiral. Here, inward-pointing hairs coax the prey toward its own digestion. Exit is possible, in theory, but never in practice. Nature's most elegant reminder that direction is destiny. Each of these traps may be active or passive, a distinction not unlike the difference between thought and instinct. Some, like the sundews, flex and bend, their tentacles curling to enclose the doomed. Others, such as Triphophilium, merely wait, silent and sure, their stickiness enough to satisfy their hunger. These exemplary plants may exhibit these traps in isolation or concert, creating a symphony of consumption. Each trap is a philosophy, each mechanism a story of survival. What evolution proves, intellect must learn. Think deeply and consume completely. Now, let us learn more of the most ancient and intricate of these designs, the pitfall trap. Its genius lies not in motion, but in patience. Imagine it, an inward chamber born from a simple, rolled leaf, sealing upon itself like a secret. Nectar glistens on its rim, light filters through its pigments, anthocyanins mimicking flowers, and the prey, deceived by beauty, descends. The fall is final. The walls are slick with waxy flakes. Escape is as futile as unlearning. Below, enzymes, or sometimes whole bacterial ecosystems, break the prey into their component parts. Such structures have evolved 6 times independently, each a case of convergent brilliance. Saracinaceae, Nepenthes, Cephalatoaceae, Bromeliaceae, Families with no shared ancestor, yet arriving at the same solution. When the environment denies, evolution repeats itself until it is fed. Helia amphora, the marsh pitcher plant, is simplicity itself, a leaf rolled, sealed, and then left open to the sky. It grows in the perpetual rain of South America's tepuis, and like all thinkers drowning in excess, it requires a drainage system. Evolution obliged, a slit in its side, a sinkhole for rain. In Sarracenia, the problem of overflow is solved by the operculum, a lid flared and cunning. Some species lace their nectar with choanine, the same toxin found in hemlock. Intoxication, even in nature, has its uses. The prey falls, drunk, into its own lesson. The cobra plant, Darlingtonia californica, perfects deception. Its operculum is bulbous, windowed with false light, tricking its prey toward escape that does not exist. Ants, the internal empiricists, tire themselves seeking exit, only to fall deeper. Darlingtonia's fishtails, the forked extensions at its mouth, are architectural seductions, guiding entry. Even its juveniles mimic this form, a touch of neoteny that binds innocence to strategy. Then come the tropical nepenthes, the monkey cups. Each pitcher is born at the end of a tendril, the mid-ribs ambition stretched to its logical conclusion. Their appetite scales with their size. Some dine on insects, others on lizards, small mammals, even birds. Nepenthes bicolcarata bears 2 thorns at the entrance, an invitation that doubles as a trap, an exquisite paradox of danger and desire. The Albany pitcher plant, Cephalotus follicularis, takes another form. Its moccasin-like pitchers catch rain and prey alike. Its rim bristles with barbed overhangs, a clever adaptation that prevents escape. And finally, Brocchinia reducta, the bromeliad that traded sunlight for shadow. Its waxy leaves form a deep urn, an aquatic grave where insects drown, and sometimes frogs take shelter. Through death, the cycle of life persists. Now, let us consider the flypaper trap, the art of stillness. Where the pitfall relies on gravity, the flypaper relies on the gentle tyranny of adhesion. It does not chase its prey. It invites proximity and then never releases it. These leaves are studded with glands, minute, glistening organs that exude mucilage. To the unsuspecting insect, it gleams like dew. To the plant, it is hunger incarnate. This design, elegant in its simplicity, has evolved at least 5 times across the plant kingdom. Proof that even in evolution, some temptations are too efficient to resist. The butterworts, Pinguicula, seem harmless enough. Their leaves shine with a soft, oily luster, the butter of their name. But that gloss conceals an exquisite deceit. A fly lands and is caught. The leaf responds not with movement, but with growth. A slow, thigmotropic curl, deliberate as thought, forms a shallow basin. The prey is held in place, safe from rain or reprieve, and digestion begins. The sundews, Drosera, are more expressive in their appetite. Over 100 species, spanning nearly every continent except Antarctica, each bearing long, mobile tentacles that end in jeweled glands. They glisten in sunlight, bait for the unwary. When touched, they bend. Drosera bermani can bend a full 180 degrees in under a minute, an astonishing speed for a plant, though perhaps not for hunger. Each motion is chemical and electrical, acid growth elongating the cells on one side, pulling the tentacle inward until it embraces its meal. Australia is the bastion of diversity, home to the pygmy and tuberous sundews, small miracles of adaptation that estivate through the dry months. They are so specialized in their diet that they have abandoned the enzyme nitrate reductase, for they no longer need the soil. Their nitrogen walks to them. The Portuguese dewy pine, Drosophilum, is kin in form, but not in temperament. It is passive. Its leaves cannot move. It gleams under desert sun, thriving where carnivory should not exist. To hunger and endure beneath aridity, this is intelligence of a higher order. In Australia, the Biblis, or rainbow plants, shine with similar intent. And then there is Triphyphyllum peltatum, a mystery among climbers. Most of its life it ascends as a liana, seeking the sun, but in its youth, it remembers hunger. It turns carnivorous, secreting mucilage to trap prey and harvest the nutrients required to one day bloom. Hunger, then, is not always permanent. Sometimes it is a phase, a necessary act of consumption to ensure eventual flowering. A metaphor not lost on either nature nor intellect. Note how these genera align: Droseraceae, Nepenthes, Ancestrocladaceae, Dioncophilisae, Plumbaginaceae. An evolutionary congress bound by the chemistry of predation and pigment. Down to the molecular level, their lineage is written in shared molecules. Plumbagan, an echo of hunger in chemical form. Adhesion is patience turned physical. The flypaper trap is the philosopher of carnivory, motionless, luminous, irresistible. And as ever, knowledge adheres to those who hold still long enough to absorb it. Next, we shall learn of snap traps, the reflex of hunger. Observe the Venus flytrap and her aquatic kin, Aldorvanda vesiculosa, the only two beings in the plant kingdom who practice the swift art of entrapment. They are the culmination of a long evolutionary patience, descended from ancestors that once relied upon the slow seduction of sticky mucilage. But here, at last, the stillness becomes reflex. Their mechanism is exquisite. Each trap is a hinged pair of lobes, lined with delicate trigger hairs, 3 per lobe in Dionea muscipula, and many more in Aldrovanda. A single brush means nothing, but 2 touches, within half a minute, awaken the plant. Ions surge, electrical signals race along the midrib, and tension, so carefully held, releases in an instant. The leaf inverts its shape, snapping from convex to concave. The prey, once curious, now finds itself imprisoned in a living geometry. This is thigmonasty, or seismonasty, movement without direction, yet full of purpose. Should the trapped creature struggle, its panic only deepens the plant's resolve, each twitch pressing the lobes tighter, sealing a hermetic chamber. Within this chamber, the enzymes awaken. Digestion begins. Over the course of a week or 2, the plant consumes what it needs: nitrogen, calcium, the essence of life itself. Then, when the meal is complete, the trap reopens. The husk of what was once alive remains, ghostly, until rain or time carries it away. Each trap may repeat this ritual 3 or 4 times before it dulls to the touch, its sensitivity lost, its work complete. Like any living instrument, even the most precise mechanism fatigues from repetition. Let us turn now to bladder traps, the mechanics of negative pressure. Bladder traps belong solely to the genus Utricularia, the bladderworts, plants that hunt not with movement or light, or even mucilage, but with vacuum. Each bladder, or vesicula, continuously pumps ions outward, drawing water with them by osmosis. The result is a chamber held in a partial state of vacuum, its delicate walls bowed inward, waiting, hungry. At the mouth of each bladder rests a hinged door, no thicker than a membrane, and guarded by a pair of trigger hairs. In aquatic species, these hairs act as levers. When a minute invertebrate, Daphnia, for instance, brushes against them, the door flexes, the seal breaks, and the surrounding water surges inward, carrying the creature into the hollow in a rush of fluid and finality. The trap reseals instantly, and digestion begins. Some bladderworts, such as Utricularia sandersoni, live not in open water, but in saturated soil. Their traps are triggered differently, yet the principle remains the same: suction, enclosure, dissolution. In colder climates, aquatic bladderworts retreat into dormancy. They die back to compact resting bodies known as turions, awaiting the return of warmth. Others, like Utricularia macrorhiza, appear to regulate the number of bladders they produce in response to the nutrient richness of their environment, an elegant economy of energy and need. From the elegant vacuum of Utricularia to the labyrinthine channels of Genelicia, carnivorous plants continue to refine the art of capture. Lastly, we proceed to lobster pot traps, the spiraling labyrinths of hunger. The lobster pot trap, sometimes called a corkscrew trap, functions through directional architecture rather than force. It is a chamber easily entered, yet nearly impossible to exit. Its passage is guarded by inward-pointing hairs that compel prey ever inward. This mechanism reaches its most refined expression in the genus Gynlicea. The corkscrew plants, small, soil-dwelling carnivores specialized in capturing aquatic protozoa. Their traps are highly modified leaves, Y-shaped in structure. The two upper arms of the Y form spiral entryways, coiling downward into the earth. Once a protozoan or other microscopic prey enters these channels, the inward-pointing hairs prevent retreat, guiding it inexorably toward the lower arm of the Y, an internal chamber where digestion takes place. Water currents, likely generated by osmotic pumping similar to that of Utricularia, are thought to aid in directing prey deeper into the trap. This shared physiology suggests a close evolutionary relationship between bladderworts and corkscrew plants, two expressions of suction-based predation, one dynamic, the other architectural. Beyond Gynlicea, trap forms reminiscent of the lobster pot occur in other lineages, most notably Sarracenia, Satancia, Darlingtonia, Californica, and, according to some horticulturalists, Nepenthes astrilocotes. In each, we see convergent echoes of the same principle, a one-way path that rewards curiosity with dissolution. Our next domain of interest is how carnivorous plants are the alchemists of appetite. Within their tissues lie specialized multicellular glands, organs of secretion and dissolution. These glands exude digestive fluids that both immobilize and disassemble prey, reducing once living matter to elemental nourishment. The process is not unlike thought itself, the external made internal, complexity rendered soluble. In species that rely on adhesive traps, such as sundews and butterworts, the digestive secretions are laced with saccharides, viscous, sweet, and lethal. In others, particularly among the Nepenthes, the fluid is a clear solution enriched with potassium, sodium, calcium, and magnesium ions, along with a complex proteomic suite unique to each genus. Peroxidases in some species further catalyze the oxidative breakdown of organic material, while hydrolytic enzymes, proteases, chitinases, phosphatases, and nucleases render proteins, exoskeletons, and nucleic acids into absorbable forms. These compounds may be stored in subcellular compartments or synthesized anew for each act of digestion, an elegant efficiency of resource allocation. Each droplet of enzyme is an act of precision. Too much, and the plant wastes energy. Too little, and the prey's promise remains unrealized. The evolution of carnivory was explored by Charles Darwin, ever the patient observer, who devoted 16 years to cultivating and experimenting with carnivorous plants in his Kent greenhouse. Darwin noted with characteristic precision that Utricularia and Nepenthes bore no true relation to Droseraceae, and thus must have evolved their predatory habits independently. He was correct. Modern molecular evidence confirms that carnivory is not the product of a single ancestral innovation, but of at least 6 distinct evolutionary convergences. The mechanisms, pitcher traps, fly papers, bladders, snaps, and lobster pots, are analogous, not homologous. Different answers to the same question posed by ecological scarcity. Independent origins of carnivory have been identified across multiple orders of angiosperms. The earliest of these lineages dates back some 85 million years. The youngest, Brocchinia reducta, emerged only 1.9 million years ago, a blink in evolutionary time. The fossil record offers little evidence of this transformation. Carnivorous plants, being primarily herbaceous, leave few traces of their delicate architecture. Seeds and pollen are all that remain, an evolutionary dialogue written mostly by absence. Recent genomic analyses, however, illuminate what the fossils cannot. Carnivory did not require the invention of new genes, but the repurposing of old ones. Genes once tasked with defense, wound repair, or nutrient transport are now refined into instruments of predation. Evolution, it seems, is an act of reorganization rather than creation ex nihilo, an editing of intent within the lexicon of life. This is evidenced by the fact that most carnivorous plants inhabit environments marked by intense sunlight, waterlogged soils, and acute deficiencies of nitrogen and phosphorus. These nutrient constraints provide the ecological impetus for deriving nitrogen from an alternative source: prey. The abundance of light permits a trade-off between photosynthetically efficient foliage and photosynthetically costly traps. To offset the energy lost to these modified structures, the nutrients gained through carnivory must enhance photosynthesis indirectly by enabling greater investment in total leaf mass and growth. Thus, under conditions of nutrient scarcity, with adequate light and water, the capture and digestion of prey yield the greatest photosynthetic returns. It is under these special ecological circumstances that the evolution of increasingly effective and efficient carnivorous adaptations is most strongly favored. Given the substantial energetic demands of carnivory, encompassing the synthesis of lures, digestive enzymes, and specialized leaf morphologies, along with reduced photosynthetic efficiency per unit of leaf area, some researchers propose that carnivory represents an evolutionary ultima ratio, a last resort strategy that arises only when nitrogen and phosphorus availability falls below the threshold compatible with purely autotrophic life. And yet, in this biological extremity lies a mirror of the human mind itself, ever reaching beyond sufficiency, compelled to consume what nourishes thought. Just as these plants evolved intricate mechanisms to secure their survival, so too does human cognition fashion traps for understanding, ingenious constructions through which information is captured, digested, and transformed into knowledge. It is to these mechanisms and the lessons they infer that we now turn. Though the fossil record of carnivorous plants is scant, much can be gleaned from the design of their extant traps and the ecological roles they fulfill. Current theory proposes that botanical carnivory arose in profoundly nutrient-poor environments, dictated by a delicate cost-benefit balance. In such contexts, an organism's energy is finite. Every allocation must repay itself in survival and reproductive advantage. Carnivory, then, could endure only if the nutrients obtained from captured prey exceeded the metabolic cost of constructing and maintaining the trap. Morphological and molecular evidence reveal that these adaptations emerged independently through convergent evolution. Pitfall traps, for example, arise from the rolling of leaf margins, as in Sarracenia. The vascular architecture of the trap's keel, with bundles oriented in opposing directions, supports the fusion of adaxial leaf surfaces. Flypaper traps follow a similar evolutionary narrative, progressing from sticky yet non-carnivorous leaves to passive adhesive surfaces, and finally to motile active forms. Molecular phylogenetics shows that the lineage of Dionaea and Aldrovanda diverged from Drosera, transforming active flypaper mechanisms into the rapid snap traps of today, a striking refinement from adhesion to mechanical predation. It is suggested that all trap types share a common ancestry. Such leaves retain rainwater, fostering bacterial growth. Insects landing on these leaves are ensnared by surface tension and suffocate. Bacteria accelerate decomposition, releasing nutrients that the plant absorbs through its foliar surface, a process mirrored in many non-carnivorous species. Leaves that excelled at retaining insects or water gained a selective advantage. Cupped leaves might have evolved into pitfall traps as margins deepened and hairs were reduced, while stickier leaves, producing mucilage, gave rise to flypaper traps. Some traps defy this pattern. Carnivorous bromeliads, for instance, Brocchinia and Catopsis, repurpose the urn, a structure present in all bromeliads, supplementing it with waxes and other adaptations to ensnare prey. Lobster pot traps, such as those of Genlicea, remain enigmatic. They may have evolved from bifurcated pitchers targeting terrestrial prey, or from expanded prey-guiding protrusions seen in bladderworts. Their helical form maximizes trapping surface in all directions when hidden beneath moss. Bladderworts offer yet another innovation. Terrestrial pitchers prevent escape by wax, gravity, and narrow tubes. Submerged pitchers, however, allow swimming prey to flee. In Utricularia, a one-way lid likely evolved into the bladder's door, later made active by creating a partial vacuum, tripped when the prey brushed the trigger hairs. Active glue traps employ rapid movement to secure prey. Such motion arises either from growth or from turgor changes that swiftly alter cell volume. Slow-moving flypapers, like Pinguicula, rely on growth. The Venus flytrap uses turgor to eliminate glue entirely. Stalked glands that once exuded adhesive material became teeth and trigger hairs in snap traps, a masterful example of natural selection repurposing existing structures for new, lethal precision. Botanical carnivory has emerged independently across multiple angiosperm families, scattered through the phylogeny. This repeated evolution demonstrates convergent solutions, disparate lineages crafting remarkably similar traps and feeding strategies. Genetic analyses, such as those published in 2017, reveal astonishing convergence at the molecular level. A digestive enzyme exhibiting the same functional mutations across unrelated lineages. Carnivory, in essence, exists on a spectrum, as does all curiosity, which brings us to the subject of the proto-carnivores. To be truly carnivorous, a plant must first possess at least one trait specifically adapted for the attraction, capture, or digestion of prey. Only a single evolved adaptation suffices, as many extant carnivorous genera lack one or more classical features. Second, the plant must absorb nutrients from their captured prey, translating them into measurable fitness gains. Enhanced growth, pollen production, or seed set. Opportunistic nutrient use without prey capture does not meet this standard, nor do defensive traits that kill insects without explicit nutritional benefit. The boundaries blur further. Some carnivorous plants lack endogenous digestive enzymes and instead cultivate mutualistic or symbiotic relationships with bacteria or insects. Modern definitions of carnivory accommodate these interactions, yet ambiguity remains. Certain plants appear carnivorous, meeting some, but not all, criteria, illustrating the continuum from wholly non-carnivorous forms to borderline predators to simple, unspecialized traps like Helia amphora, culminating in highly refined mechanisms, as seen in the Venus flytrap. Consider the genus Rhoridula. Sticky, resin-tipped leaves capture insects with precision, yet the plant itself does not digest the prey. Instead, assassin bugs consume the trapped insects, and the plant absorbs nutrients from their leavings. By some definitions, this still constitutes carnivory. Other candidates reside in ambiguity. Members of Mardiniaceae, such as Ibicella lutea, feature sticky leaves that ensnare insects, but conclusive evidence of nutrient absorption is lacking. Seeds of shepherd's purse, urns of Papalanthus bromeliadoides, bracts of Placiphora fetida, and flower stalks or sepals of Stylidium capture and kill prey, yet their classification remains contentious. Even liverworts, such as Cloeura and Pleurazia, display sac-shaped leaves that trap ciliates, potentially digesting them, a remarkable miniature expression of botanical predation. And exceptions abound. Nepenthes ampullaria, a pitcher plant, has forsaken active carnivory, instead capturing falling leaves to nourish itself, an elegant deviation demonstrating that evolutionary trajectories are not linear and that carnivory is a versatile, context-dependent strategy. That complexity has allowed these stunning specimens to proliferate as widely and dynamically as thought itself. This leads us to the domain of ecology and the modeling of carnivory. Carnivorous plants are widespread, yet rare. They thrive in extreme habitats, bogs, marshes, even deserts, where sunlight and water are abundant, nutrients are few, and competition is minimal. Only under such austere conditions does carnivory pay, allowing these exquisite predators to justify the energetic costs of their traps. The Venus flytrap epitomizes the archetype, enduring soils almost devoid of nitrate and calcium. Nitrogen is essential for proteins, calcium for cell walls, phosphate for nucleic acids, and iron and magnesium for chlorophyll. Yet the bog is acidic and ammonium-rich, a potent double-edged sword, usable in trace amounts, toxic in excess. Even so, bright light and constant moisture permit growth, while sphagnum mosses compete minimally. Exceptions exist. Drosophilum lusitanisum persists at desert edges, and Pinguicula vallisneriflorea clings to calcium-rich cliffs. Carnivory compensates for nutrient deficiencies, providing nitrogen, phosphorus, and occasionally potassium. Dependency varies. Pygmy sundews cannot use soil nitrate. Butterworts favor a mix of inorganic and organic nitrogen, and European bladderworts utilize both equally. Prey supplements the soil's deficiencies, allowing photosynthesis and growth to continue. Leaves are dual purpose, intercepting light for photosynthesis and serving as traps. Photosynthesis captures solar energy to digest carbon dioxide, forming sugars and biomass while releasing oxygen. Respiration, in turn, consumes energy, producing carbon dioxide. Net growth, gross photosynthesis minus respiration, defines a plant's potential. Trap modification imposes costs. Pitchers must remain upright. Flypapers produce glue. Snap traps expend adenosine triphosphate to trigger. As photosynthetic efficiency decreases, respiration rises and growth potential narrows. Carnivory, then, is a costly gamble. Worthwhile only when prey-derived nutrients improve photosynthesis, particularly through ribulose 1,5-biphosphate carboxylase oxygenase. The nitrogen-rich enzyme crucial for carbon fixation. The spectrum of carnivory is evident. Full-time, energetic predators like the Venus flytrap invest heavily in snap traps. Partial predators like Tryphophyllum petaltatum employ simpler, slower, flypaper traps. Energy expended on trap construction measures carnivory. The more investment, the greater the adaptation, but also the higher the cost. Modeling carnivory reveals a delicate balance. In sunny, nutrient-poor habitats, Moderate investment optimizes net photosynthesis. Too little, and growth falters. Too much, and light or carbon dioxide become limiting. In nutrient-rich but dim habitats, carnivory offers no advantage, and zero investment is optimal. The degree of carnivory correlates with habitat extremity. The rarer the nutrients, and the more generous the light, the more pronounced the adaptations. Seasonal shifts illustrate this pragmatism. Sarracenia produces flat, non-carnivorous phyllodes in winter when light wanes and insects are scarce. Tuberous sundews retreat to tubers in dry seasons. Bladderworts to turions in winter. Some plants, like Triphophillium peltatum, vary carnivory based on life stage nutrient needs, particularly potassium, before flowering. Carnivorous plants are poor competitors outside of specialized habitats, succeeding only where others fail, nutrient analogues to cacti in deserts. Bromeliads demonstrate pre-adaptation. Their urns collect nutrients, yet most epiphytic species remain shaded and non-carnivorous. Only ground-dwelling species, like Brocchinia reducta, express true carnivory. Fire ecology and habitat disturbance often dictate survival, shaping distribution and abundance. Ecologically, carnivorous plants foster remarkable communities. Sundews, pitcher plants, and bladderworts support insects, frogs, spiders, and even small mammals. Nepenthes raja benefits from tree shrews and summit rats, which feed on secretions and defecate into the trap, a particular form of nutrient acquisition. Bats, mosquito larvae, and myriad endophytes inhabit or exploit these microhabitats, forming complex, mutualistic, commensal, and kleptoparasitic networks. Pollination introduces another layer of tension. Carnivorous plants must balance prey capture with reproductive success, avoiding the pollinator-prey conflict. Strategies vary. Spatial or temporal separation of flowers and traps, targeting distinct insect guilds, or offsetting short-term reproductive losses with long-term growth gains. Ultimately, carnivory is neither ubiquitous nor effortless. It is a precise orchestration of energy, habitat, and opportunity, a seductive negotiation between plant, prey, and environment, played out across bogs, cliffs, deserts, and trees, where only the most finely tuned adaptations endure. Carnivory, then, is not merely a curiosity of nature, but a negotiation, a delicate arithmetic of scarcity and adaptation. Each trap, enzyme, and symbiosis reflects an ancient calculus of survival, performed in the margins where other flora cannot endure. Yet even these specialists, evolved for extremity, find themselves imperiled by the extremities of human alteration. As we turn to the question of conservation, we must ask, what becomes of a creature perfected for poverty when abundance, our kind of abundance, remakes the world in its own image? Of the carnivorous plants assessed by the International Union for Conservation of Nature, nearly half stand upon the threshold of disappearance, classified as vulnerable, endangered, or critically so. Their decline mirrors that of their habitats, wetlands drained for agriculture, moss beds paved over for housing, and nutrient-poor bogs dangerously enriched by our excess. The same conditions that made carnivory possible, low nutrients, still waters, slow decay, are those most easily undone by human ambition. Across continents, the pattern repeats. 19 species in North America, 15 in Asia, fewer but no less imperiled elsewhere. Their predators are not insects, but industry, development, pollution, invasive species, and the silent violence of climate shift. Even among the carnivores, kinship defines fate. Species of a shared genus often fall together, as if bound by evolutionary memory. And yet, where the conditions of their survival are recreated, they return. Bogs can be reborn. Seeds long dormant will stir in restored light. To conserve the carnivorous plant is to conserve a philosophy of endurance, that life, even when starved, invents its own sustenance. In preserving them, we preserve the exquisite intelligence of adaptation itself, proof that beauty and ferocity may coexist, that fragility need not mean defeat. What, after all, is carnivory but curiosity made flesh, made leaf, a reaching beyond constraint, an intellect expressed through appetite? These plants do not consume for cruelty, nor kill for conquest. They reason chemically. They think in enzymes and lures, in scent and symmetry. In them we glimpse our own reflection, the ache to endure, the compulsion to know, the impulse to turn limitation into design. Their genius lies not in predation, but in adaptation, the quiet insistence that even in scarcity, life persists. So when we preserve them, we are not merely saving a species, we are safeguarding a syntax of survival, an intelligence written in chlorophyll. And perhaps, in doing so, we remind ourselves that thought, too, is a kind of hunger, and that to feed it wisely is the most adaptive act of all. You have been listening to The Soul of Curiosity, a study sustained by patrons and students like you, whose support makes inquiry itself possible. If you found nourishment in these reflections, like, subscribe, leave a comment, and consider joining those who keep this work alive. Every contribution, every conversation, feeds a mind's unending appetite. Until next time, obey your curiosity and walk wisely. A very special thanks to my supporters, Cactus Cutie, Kimbop, Mr. Boo, and Tom Staz.